Hi, everybody. Well, we're back, and it's time to talk about the biology of mind and consciousness. For this segment, I'm going to cover the topic of neurons, which I like to start with this. I like the structure of our textbook because the first real content chapter, talking about the psychological concept, not just arguing for the science of psychology, but starting with the real concept of psychology, with chapter 2, we have the smallest things that can possibly contribute to your behaviors and your mental processes. The neurons that make up your brain and your spinal cord, and then the chemicals that communicate between the neurons that allow those neurons to share information. So we're starting with the very smallest things that could possibly contribute to your behavior and mental processes. And way later in the text, we're going to talk about the biggest things that could impact your behavior, culture, and your social setting, and things like that. So I kind of like the structure of this book because it sort of takes us through from the smallest possible contributors to the biggest possible contributors, everything in between. So let's get started with the biology of mind and consciousness. Well, this little diagram is illustrating a neuron. Now, if you ever get the chance to see a nice stained microscopic image of a neuron, they don't really look very much like what you're seeing right here. This is very neat and orderly, believe it or not. Depending on how the slide is stained, it could be a neuron could look significantly different under a microscope. So this is a good cartoon depiction showing you the structures that may or may not look exactly like this in real life. But it's basically this kind of cell. And what's very likely is that you've had some kind of biology class that has taught you about different kinds of cells, plant cells, skin cells, different living organisms cells. And they all share something in common. They all have a cell body that supports the life of the cell. So in the nucleus of the cell body is the DNA and the RNA and all the energy generating mitochondria and all that kind of thing. So all cells have that in common. They all have cell membranes, things like that. What makes a neuron really unique is all this stuff that's sticking out of the neuron. So we have on the cell body a lot of what they call processes, which are these branching fibers that stick out from the cell body, looking very much like roots or like a tree branch with no leaves on it. These are called dendrites. Dendrite in Latin, I think it means roots, but it might mean branch. I can't remember. But you know what? In Latin, the word means exactly what this thing looks like, which is these branching fibers. The job of the dendrite is to collect information and carry it into the cell so that the cell then can process the information. Dendrites are designed to collect information. Cell bodies themselves, the membrane of the cell body, can actually directly collect information. You don't need dendrites to be a neuron. But a neuron that does have dendrites has the ability to grow those dendrites out and reach towards other cells so that they can collect information from cells that can be pretty far away from the neuron itself. Dendrites have been known to get, several centimeters long. In fact, they can be really long, going down your spinal cord. So dendrites they grow. They're not fixed in size. You're not required to have any dendrites on a neuron, and you're not limited to a certain number of dendrites that you can have on a neuron. You can have any number that the neuron feels the need to grow. Sometimes dendrites are kind of called to grow by neighboring cells. Neighboring cells are sending out chemical messages that let that cell body know, you need to grow a dendrite and reach out to me because I have some stuff I want to tell you, so grow out here so I can touch you and I can tell you. So they grow all the time. In fact, they've seen evidence that dendrites grow through our entire lifespan. They accidentally discovered that. When I was in school back in the dark ages of the 1980s, when I was in college, we were told that once you reached adulthood, your brain was basically just atrophying from that point forward, that there was no new growth in the brain, and that, in fact, the brain shrinks in size with every year that you are alive. You are homing in on just the neurons that you're using, and you're eliminating the ones that are excess. So that was what I was trained in, and it wasn't until the late 90s that researchers accidentally discovered in a patient who was being treated for brain cancer, they put a marker into his bloodstream so that the marker would attach to any new growth that was going on, and then they would be able to use a CAT scan type of machine and look for evidence of where that marker had attached itself, and then they would know where there might be more tumor growth. And they discovered that it was attaching itself to dendrites in the brain of this patient, and that these were new dendrites that were growing. And this patient was 80 years old. So, and had cancer, right? So this is the person who's, you might think, well, stressful situations, maybe you're not developing new growth, and you're an older person. I thought you weren't supposed to be making new brain connections and things. And it turns out that that case caused people to rethink how the brain actually develops over the lifespan. So dendrites now, we think, grow your entire lifespan. And lots of other researchers have replicated what those initial researchers accidentally found. So dendrites are always growing, always collecting information. That's their job. They take that information, they conduct it to the cell body. And the cell body decides whether that information is worthy of being passed on. If the cell body decides that it's worthy of being passed on, it's going to use the axon to send that message to neighboring cells. So there are limits on axons. You have to have an axon to be a neuron, and you can only have one axon if you're a neuron. If one was malformed and had two axons, it would literally implode. They've caught it on film where the little cell body just goes boop and just explodes, and the body reabsorbs it because it was not going to work right. You can only have one axon. All the information goes down the one axon. Now in this picture, you see these nice white bubbles that are painted over the axon, and they've cut it away in that one spot so we can see the axon. Those white bubbles are representing lipids, fatty deposits, that in case some of our axons, most of our axons, are myelinated. It's called a myelin sheath, and its function is the speed transmission of the message from the cell body out to the neighboring cell. If you think about the myelin sheath being like the coating that is on the electrical wires in your house, right? I mean, look at, I'm talking on a computer, and I see my computer's cable, and it's covered in what appears to me to be plastic. I don't know what it really is. And I know inside the black plastic that is the cable that I'm using, it would be wires, metal wires that can carry electricity from the wall to my computer. The reason why it's covered in that plastic is because if it weren't there, the electricity would jump off of the wire, maybe catch my house on fire. That's not good. It could jump off the wire and not provide enough electricity to the computer. If it's being disseminated off the wire, the whole length of this, I think my wire is probably 6 feet long. If it's being disseminated off the wire for that entire 6 feet, by the time you get to the end, where it's supposed to be powering the computer, it may be out of oomph. It's lost enough of the electricity that it can't actually power the computer. When I was in grad school, I lived in a house that had old-fashioned 1940s knob and tube wiring, and basically the wires are bare and go around the porcelain knobs that are up in the attic. And only when it goes down through the wall, it has a little bit of asbestos insulation to keep the electricity from catching the house on fire. But it wasn't really the kind of insulating wire that we have now, the coating that we have on our wires now. And it really illustrated for me the importance of keeping the electricity on the wire because, that house, for one thing, didn't have very many outlets. It was a 1940s house. But for another thing, it had that knob and tube wiring. So on the same circuit was the kitchen and the bathroom. And if my husband was cooking a burrito in the microwave, I couldn't run the hairdryer in the bathroom. Like there was not enough power making it to the bathroom while he was microwaving. And that was in part because there just wasn't enough power, but it was also in part because the electricity was actually leaving the wire, so that when it got to the microwave, it probably didn't have as many amps as it had when it left the box. And then once it passed through the microwave area and was supposed to come to the bathroom, it absolutely did not have enough power. That could happen to us in our minds if we don't have enough myelin coating. And we know that this happens from people who have problems with their myelin sheaths. People with multiple sclerosis, what that word actually refers to, the sclerosis, means hardening, and it's referring to the myelin sheath. So in that disease, the disease is actually attacking the myelin coating, and it usually affects the distal regions first. Distal means farthest away from the brain. So feet are the first to go numb or lose, control, so you can't like point your toe or wiggle your toes as well when you're having this hardening. And then as the disease progresses, it can move closer and closer into the proximal areas. Then you can end up with difficulty breathing and other kinds of really dramatic effects. And it's happening because the myelin sheath gets rigid. And a rigid myelin sheath does not insulate as well as a flexible, fatty myelin sheath does. So when we look at people who have problems with their myelin sheath, we can see the impact, how important myelin is to ensuring that messages get passed on. All right, I have one last little soapbox thing to tell you. Myelin sheaths are made up of fat, and they are laid down in the first 3 years of life. So it's really super important that infants and toddlers get adequate fat in their diet. So they need whole milk. They need mom's milk. Mom's milk has the highest fat content of all the mammals. I think the pilot whale makes slightly fattier milk than a human, but other than that, we have the fattiest milk, and it's largely because it contains the kinds of fat that are really easy to lay down in the brain. So we have this perfect balance, and fatty cow's milk is good for toddlers who can consume it, who aren't allergic or don't have a problem with lactose, because it also has the kind of fat that lays down nicely in the brain. There are other ways, other things that people can eat to make sure that they're getting the right kind of fat that'll lay down and make that myelin sheath. If you miss the opportunity to lay down that myelin sheath, it's not like you can do it later. And we see this in children who were, during that critical period when they needed to be laying down this myelin sheath, and there was a famine in their area, and they didn't get adequate fat and dietary cholesterol and the things that you need to set up the brain. And for the rest of their lives, they have slower reaction times and things like that. So it's a very important thing that we make sure that we follow proper dietary practices to make sure that our children lay down the myelin sheath that they need. The last part of the structure is called the terminal branches. The terminal branches of the axon. And you'll notice it looks like a hand almost with sort of buttons on the end of the fingers. The terminal branches make contact with the neighboring cell so that it turns out that your single axon can communicate with multiple cells. Each of those terminal branches could be touching a dendrite from a different cell. So each of those terminal fibers could be, so we end up with, 20 on this picture, so I haven't counted it, opportunities to communicate with the neighboring cell. So terminal branches are the thing that allows this individual neuron to pass its message to a lot of other neurons. All right, here's a nice cross-cut of actual axons. This has been colorized, so we can tell the difference between what has been colorized to look a little like orangey in the middle. It kind of looks like, I don't know, this to me looks like dog treats, right? There's those dog treats that you can get. They have some kind of filling in it that tastes good to the dog, and the crunchy outside that kind of brushes their teeth. That's what it looks like to me. The inside that would taste good to the dog, that would be the actual axon, the fibers that are going to carry the information. And then that yellowish, flaky stuff around the outside, that's your myelin coating. So when we zoom in close enough, it looks a lot different than that picture looked. And you'll notice that this is a bundle of axons. This is not a single neuron. This is a bundle, and it's actually a cross-section of what we would call a nerve, where you've got a bundle of a bunch of axons all traveling together to some location. This could be the optic nerve carrying information from the eyes to the brain. It could be a nerve that's going to go out and move your thumb. I mean, I'm not sure where this came from exactly in the body, but it's a cross-section of a bundle of axons. All right, so I keep talking about sharing information. And that's a little vague when you're talking about communications between cells, because you're like, what kind of information can you possibly be passing on? Well, it's electrical. Our neurons, from the tip of the dendrite to the tip of the terminal fibers, within that distance between the neuron, that is entirely an electrical object. So all communication is going to be electronic, or electrical, I think is the correct word. Now this little diagram is showing you how it's electric. Inside the axon, when it's resting, is negative polarity. And when an action potential is going to occur, these what are called ion gates open up along the axon and allow positive polarity to come rushing in. And that causes an electrical current that pushes down the axon. And the ion gates open in sequence down the length of the axon, so it ensures that that electrical impulse travels down the axon. I'm simplifying quite a bit. If you really think this is super interesting and you want to know more, taking a nice physiological psychology class is a good place to go, or maybe an anatomy and physiology class might cover it. But I'm just going to kind of gloss a little bit, give you an introductory psychology level understanding that it is in fact electrical. So when we have electricity going down the axon, we call that an action potential. So here's the definition of an action potential. I think I lifted this exactly from our textbook, maybe took a couple of words out. When stimulation exceeds threshold, the cell will fire. Okay, I think we need to define a few words. What's threshold? So when your axon is at rest, when your neuron is at rest, I should say, when the neuron is resting, it's not being stimulated by its neighbor, it's not having an independent thought that it feels like sharing with its neighbor, it's just resting. That is what we call its threshold level. It has gotten accustomed to this level of stimulation. So when the incoming information is more demanding, more attention grabbing than what's been going on in the neuron so far, we call that exceeding threshold. Okay, so it's easy to talk about it when we talk about the cell was at rest and then new information came in. What about this? You are at a loud party. Music's playing, people are talking, there's, periodically people yell or there's just a bunch of noise. And you're really interested in talking to an individual at this party. So you guys have got your heads close together, you're talking into each other's ears, and you're really looking at each other so you can concentrate and actually have this conversation in this very noisy situation. Believe it or not, your brain gets used to that level of, I'll just say, din in the background, just noise that is, you don't care about it, it's just noise. You're trying to actively ignore it. Your brain gets used to it. Okay, well, this is the background noise that I don't need to be paying attention to and I can just ignore. That becomes its resting stage. So it would take incoming information that exceeded that din for you to now hear it, for your attention to be pulled by it. And you might have had an experience where you were, for example, engaged in very intent conversation with somebody, and you were able to effectively tune out all the noise going on around you until you heard somebody say your name. And that triggered an action potential. You're like, whoa, I think this is relevant to me, and it caught your attention. So the threshold doesn't mean that nothing's going on. The threshold means your cell has gotten accustomed to what's going on. For you to have a new firing by that cell, the incoming information would have to exceed what you've gotten used to, whatever the new threshold is. And then the cell will fire. So the word fire is used in place of action potential because it is like a firing of a gun or something where, it goes traveling rapidly down that axon. And you kind of can't stop it once it starts, right? Just like a firing, right? When the cell fires in an action potential, it is an all or nothing response. The cell either fires or it doesn't. There's no such thing as representing intensity by a cell firing less intensely than it usually would, or something like that. It can't do that. The cell either fires or it doesn't. For those of you who are familiar with computers, we could say it's like a binary code, right? It's either on or off. This switch is either on or off for passing this information. So how do we represent intensity? How do you know the difference between a tickle and a slap, right? Like, how do you know? Well, one way that you know is by how many cells are firing at the same time in response to that same stimulus. So a slap on your back is going to cause more cells to fire in response than a tickle on your back will activate. Fewer cells will be involved in the tickle. And then the frequency of firing. So a more intense stimulus, your cells will fire more often than a less intense stimulus. So if it's a really bright day out, all your cells are saying, oh, bright light, bright light, bright light, bright light, bright light, as fast as they can, all of them, and they're as fast as they can, bright light, bright light, bright light. When it's foggy out, you have some cells going, yeah, I think it's daytime. Yeah, I think it's daytime. So fewer cells, less often, passing the information. Of course, once you know it's daytime, they all stop telling you. Eventually, that becomes their resting level of input, and now it would take a flash of light or something to attract your attention. So you will adapt very quickly, but when it's a new stimulus, the cells pass that information along, with information about intensity being represented by number of cells and then frequency of firing. Okay. The synapse. Okay, so I told you that a neuron is completely electric. Electricity cannot reliably jump gaps. It can use the molecules of water to conduct itself. It can, use metal, things like that. But we can't count on electricity to communicate between cells because our cells have little synapses between them. So in this picture, what you're seeing is the terminal button or the terminal fiber of the sending cell. That's the bulgy thing. And then the flat thing on the bottom that's sort of curvy, that is the receiving cell. There's a little gap, and we need to cross it. So at the terminal fiber, the electrical impulse, the action potential, comes down and actually causes little vesicles, little balls containing molecules in them, to rupture out the end of the terminal fiber, float across the synapse, and lodge in the receiving cell's receptor sites. And that's how the message gets passed. So those little balls that are in the picture here, the little blue balls, they're floating across the synaptic gap. They're going to lodge in the receiving cells in little receptor sites that are half circles there waiting for exactly this molecule to show up. Once enough of those receiver cells are occupied, that'll convince the neighboring cell to have an action potential of its own. So that's how information gets passed. It's chemical when we talk between cells. Within a cell, it's electrical. Between cells, it's chemical. Now, you may see out in the world or even in our textbook different terms for this space between cells. I've got on the slide, I called it the synapse. In the picture itself, they call it the synaptic gap. And sometimes people call it the synaptic junction. I've even heard people call it the synaptic cleft. So there's lots of different words. As long as it has synapse in it, you're probably talking about the space between cells. So those chemicals that cross the synapse are called neurotransmitters. You've probably heard of neurotransmitters before, so I'm not going to belabor, what it's called and why it's called that. It's the little chemical messenger that's going to send the signal from the sending cell to the neighboring cell. That's the way that we can jump that synapse. So here I've labeled in this picture the vesicle containing the neurotransmitters. They're floating around in the terminal fiber. They're bundled up in there with little molecules all tied together. The electricity forces them out the end of the terminal fiber, and that's how they lodge on the end of the terminal fiber and then rupture because of that electrical impulse. And that's how they get out into the synapse. And then they float across in the cerebrospinal fluid that's in the brain, float across, land in the receptor site, and cause the receiving cell to have its reaction. Now here's the thing. Some neurotransmitters are what we call excitatory. They make the neighboring cell want to have its own action potential. Others are what we call inhibitory. They tell the neighboring cell, do not pass this on. The action potential stop here. This is the secret. So depending on the neurotransmitter, and then depending on the receiving cell, whether it's prepared for that neurotransmitter or not, you can be passing messages along with an action potential, or you might be actually squelching the neighboring cell from passing the information along. So let's talk about that a little bit. I have listed here just 6 neurotransmitters. So far they've identified somewhere around 100 neurotransmitters. A lot of them have nothing to do with anything psychological, so I absolutely don't know for sure how many there are. I really only know the psychological ones. And here I've listed 6 that I think will ultimately come up again in class. So I'm trying not to bog you down with a bunch that aren't going to be mentioned again in this class. So we have 2 here that are related to memory, acetylcholine and glutamate. Both of those neurotransmitters are secreted as you're learning new information, storing new information, things like that. Dopamine is a neurotransmitter that does a lot of different things. There are different pathways for neurotransmitters in your brain. There's the reward center of your brain, which is predominantly your hypothalamus. We'll talk about that more in another segment in the same chapter. Your hypothalamus is responsible for making you feel rewarded by different activities. So like sex or eating chocolate, using cocaine, things like that. When those are perceived as rewarding and you want to do them again, it's thanks to that rush of dopamine that you get in the reward center. If you look at the dopamine pathways that are more in the temporal lobes, more near your ears, those are involved, those dopamine receptors are involved in auditory information transmission, so hearing things. So while you're listening to me talk, you're having doses of dopamine secreted in your auditory cortex. Dopamine is also active in the very back of your brain in a structure called the cerebellum, and it controls movement. So there are 3 that we know of predominant dopamine pathways. So it does a lot of different things. It's a pretty basic, sort of the Swiss Army knife of neurotransmitters. It can kind of affect lots of different things. But you'll notice that they're in very different regions of the brain. So it's not like you could have dopamine secreted when you meant to, because you're experiencing something rewarding, and suddenly it makes you kick your leg because it activated the movement center. The different dopamine pathways are in vastly different areas of your brain, so they don't interfere with each other. Serotonin, we're going to talk about a lot because it is implicated in a lot of different things. It's implicated in mood. So we have, depression and anxiety being linked to low levels of serotonin. It's also active in your hunger center. There's a part of your hypothalamus that is in charge of determining whether you're hungry or not. And so one of the reasons why when we're sort of in a bad mood, we want to eat comfort foods is because when we have low levels of serotonin, one way to boost it is by eating things, right? It makes us feel better if we eat stuff. Or sleep, right? Sometimes sleep will make us feel better if we're just sort of in a what we call hypo mood, like a down mood. Sleeping can be, sleeping too much or not getting enough sleep. Sleep can be a symptom of depression, right? If you've had big changes in sleep. If you have really big changes in your eating habits, that could be a sign of depression also. So you can kind of see how this ties together. Norepinephrine has also been implicated in mood. Norepinephrine seems to elevate our moods. It also makes us more aroused, and I don't mean sexually. I mean like alert. It makes us feel more active, makes us not tired, stuff like that. And so it's been implicated in mania. GABA is they really had underestimated the importance of GABA for a really long time because it's purely inhibitory. It's one of those ones that I mentioned before where if the neighboring cell receives GABA, the basic message is don't pass this on. We're starting to realize that GABA is really super important for attention, because one of the things you have to do if you're going to pay attention to one thing is ignore other things. And so GABA helps us to ignore other things. As we age, our GABA production goes down and it becomes more difficult to tune things out. So kind of interesting things that we're learning as we're identifying more neurotransmitters and how they actually work. Now, the way that neurotransmitters communicate between cells is in what we call a lock and key mechanism. So we've got the picture of the sending cell and the receiving cell on the left, and then we have what is a kind of cartoon version of what a neurotransmitter molecule might look and what the receiving site might look like, and see how they're designed for each other. Right, there's nooks and crannies in the receptor site that will perfectly line up with the protuberances on the molecule, and when it locks together like that, it tells the brain, oh yeah, I got some neurotransmitter, I'm going to have my reaction that I'm supposed to have to this neurotransmitter. Now, there are chemicals that we can consume that can mimic those kinds of neurotransmitters. They're called agonists. Notice that the agonist is not perfectly lodging in this receptor site, but it's pretty close. And so it causes the brain to think, well, maybe this is just a malformed version of the neurotransmitter that I'm accustomed to. And so it'll have the reaction to that agonist that it would normally have to the actual neurotransmitter. So things that you might know of that are agonists, caffeine is an agonist for norepinephrine. So you get the same kinds of benefits, downsides of norepinephrine when you consume caffeine. Heroin is an agonist for endorphin molecules. And so you get the same kind of pain relief that you would get if your brain was just secreting it itself. So agonists work the exact same way as the naturally occurring neurotransmitter. The opposite of that is what we call an antagonist. Antagonists work by blocking the receptor site so that a real neurotransmitter can't get in there. Now you'll notice in this antagonist drawing, it's locking in there because it's got one protuberance that's fitting in there, but it's not activating the neighboring cell. It's just blocking the ability to communicate the message. So antagonists, for example, like aspirin products, they get into your bloodstream, they go and lock into those receptor sites in the brain that would normally receive pain messages. And so then the molecule blocks the ability to send pain messages. It's not that it takes away your pain at all, it just blocks your transmission of the pain. So antagonists just prevent the neurotransmitters from doing their normal job. All right. Normally in class, I make my students do a little knowledge test before we move on. So if you want to pause this and see if you can name all these, I'll come back and reveal the answers. Okay, so I assume you paused me if you wanted to pause me. If not, I'm about to reveal the answers. If you want to test your knowledge, last chance. All right, here we go. So just to remind you, the big part there is called the cell body. The protuberances on the cell body are called dendrites, the branching areas. That's the axon, the big long fiber. We've got the myelin sheath coating the axon. We've got the terminal branches of the axon, and then we've got this arrow indicating the action potential. So hopefully you got those all right. If not, you can still study a little bit more before you need to take a test, right? All right, well, this is a good place to stop for this section. So let's come back and we'll talk about how our nervous system is divided up.